almost at 10:05 anyway. Just one more minute. Okay, great. So welcome everybody. This is one of our journal clubs, and we are particularly thrilled to have a journal club on this wonderful paper, a Drosophila computational brain model reveals sensorimotor processing, a Nature paper, first author Philip Shiu, and I believe he may even be here right now. I'd say take it away, Archit.
All right, thank you so much, Dr. Cohen. My name's Archit. I'm a master's student at Rice University studying bioengineering. So I just wanted to present this paper, and I know Dr. Shiu is here. And if anyone else also wants to just interrupt at any point in time with any comments or questions, please feel free. So I'll start with the abstract and then go into a brief background. So this paper discusses the connectome of the Drosophila melanogaster, the fruit fly, and points out how there are, oh, I'm so sorry. I think I messed up my screen share. Is this visible?
I can see your abstract.
Okay, perfect.
Yeah, looks good.
So what this paper does is it uses the computational Drosophila connectome that was constructed earlier this year, and it tests it with leaky integrate and fire neurons to verify particular parts of sensory and motor circuits, such as feeding initiation and antennal grooming. And then it verifies those actual predictions from the computational model using optogenetic stimulation. And the purpose shows that modeling brain circuits using only synapse level connectivity with these leaky integrate and fire neurons can describe complete sensory motor transformations, which is very valuable when performing these types of studies because it's cheaper to do and provides a little bit more insight as well, looking at downstream neural connections. Now, there are various reasons to produce a computational model. Of course, there are the standard ones that it's easier to test experimental design or research questions. It's a simplified way to test stimulus generation, especially in neuroscience, when you can simply provide a stimulus using some sort of equation and show how that affects downstream reactions in the brain. It allows you to extrapolate trends to unfamiliar scenarios, and it simplifies the complexity of lab experimentation on neural tissue because it's not as necessary when you're able to do these types of tests. So essentially what a lot of the work in this paper allows for is a litmus test to see whether computational models done on connectomes are sustainable for future studies as well. And of course, we want standard considerations that it should be experimentally relevant, consistent with real world observations, relatively simple to compute, compatible with more complex scenarios, so allows for scalability and is, of course, accurate. So I'll briefly discuss types of computational models before diving into the paper itself. We know we have Hodgkin-Huxley, which was the big, many differential equations using to describe ionic dynamics within neurons. There's the Zekovich model, which is a more simplified version of that. There's leaky integrate and fire, which this paper uses, and then there's many more adaptive leaky integrate and fire, exponential integrate and fire, and so forth. The point being, they all stem from a neuron is a circuit, right? There's varying degrees of complexity in this circuit, and you can use as many variables as you want to describe what's going on. So Hodgkin-Huxley, for example, may look at inhibitory synaptic current, excitatory synaptic current, and so forth, using all sorts of ions to kind of describe this. Whereas you might want to simplify a little bit more with different types of compartmentalizations, which is what the Zekovich neuron model does, or by even simplifying it even further and just having a couple of variables like your overall voltage, your reset threshold, and so forth. Some types of behaviors aren't necessarily modeled by simplified models. For example, leaky integrate and fire models can't necessarily handle bursting behavior that you may see in certain neurons. And these types of considerations do come into play later in the paper when there are some descriptions of factors that might not be described by this simplified version of a model, but we still see good results overall, and I'll discuss those as well. So this is the equation set that the authors used in this paper for a leaky integrate and fire model. Essentially, it's just a couple of differential equations and an overall rule for your values, your synaptic conductance to change based on weights in your neurons. So we kind of just look at overall changes in voltage and identify spikes in the neurons, which allows us to represent a sort of signal. Now, all this is entirely from the construction of the connector of the Drosophila fly, and this is also in a website called Bible. There are a large production of the entire brain of a Drosophila using electron microscopy and other tools to identify exact glycation synaptic weights based on certain concentrations of different proteins in synaptic junctions, and it allows basically data of which neurons are connected to each other, as well as their overall synaptic weights, and how extremely important because it allows us to essentially stimulate certain neurons and see downstream what the effects are. So before diving into the overall methodology of this paper, it's important to mention exactly what's happening in this type of model, right? So similar to the biological brain, when you have certain neurons that are upstream and certain neurons that are downstream, essentially what happens is you have a particular set of neurons, and when they spike, they're going to send that action potential downstream to other neurons. And based on certain weights, for example, this gray neuron going to a green neuron, the weight of the synaptic connection is 80, which is double that of the weight between the gray neuron and the purple neuron, which is 40, which means that this neuron, if the gray neuron spikes, it's going to have a much greater effect on the green neuron than it will on the purple neuron. But overall, this allows for us to kind of see what sorts of neurons are important for other neurons to fire. And overall, that leads to these graphs that we've seen in regular EEG or other sorts of electrocorticography studies, and you just see, spiking of a neuron. So specifically to analyze this, the authors focused first on the Drosophila feeding network. The reason for this is that there aren't as many downstream connections that are going through, for example, the spinal tract or anything like this that hasn't been identified in Flywire. This is all something that we can model completely within the brain. And this feeding network, what it does is it focuses on a couple of gustatory sensory neurons, such as sugar sensing, water sensing, a couple of other ones like bitter, and it allows for motor neurons to move the proboscis of the fly toward that stimulus. So for example, they initially focused on sugar presentation, and they discussed how unilateral sugar presentation causes the proboscis to extend towards the side of the fly with the sugar. And we're only focusing unilaterally, and in this case, the authors focus first on the left hemisphere. In the supplemental material, you can see data for the right hemisphere as well. But focusing first on the left hemisphere of basically the fly head where sugar is presented, and seeing how the fly kind of moves its proboscis towards that sugar, towards that area. So there's a bit of a complicated circuit here, but it's actually perfect for analyzing the efficiency of this connectome because all of these neurons have been recorded, all of their weights are present in the connectome, and you can essentially copy-paste these into a leaky integrate and fire network to analyze how certain upstream stimulations, for example, stimulating a particular neuron that's sensory, a sugar neuron, can show downstream activations. For example, in this specific neuron, motor neuron 9. The reason they focus on motor neuron 9 is because motor neuron 9 is required for moving the rostrum of the fly head toward that sugar. And that's one of the most important parts, essentially, of the overall movement toward the sugar or any other gustatory sense. So focusing on this neuron allows for an overall picture of what's going on in this particular circuit. Now, based on the computational model, by taking all of these leaky integrate and fire neurons that represent the overall connectome and giving them their weights and constructing that network, we were able to identify circuit contralaterality. And that specifically means that you were able to see how pointing out sugar on the left side of that fly allows for ipsilateral, which is the same side movement, and contralateral, which is the opposite side movement, or stimulation of the motor neuron. So we see that given the computational model, that the contralateral motor neuron is far more active, has a higher firing rate, given the sugar neuron's firing rate, which is something that you stimulate in the actual network itself. And this is pretty much in line with experimental studies as well, which show that the contralateral motor neuron tends to be slightly more active than the ipsilateral motor neuron. Also, what's extremely important to note is that this study was again conducted by shuffling the synaptic weights of the network. And shuffling the weights led to a 99% reduction in motor neuron 9 activation. What that means is that previously 100% of motor neuron activation was observed, but after shuffling the weights, only 1% was observed, which shows that the weights of these neural networks are extremely important. And that's something that we already knew, but having that data through Flywire allows for this to be done now as opposed to previous cases. Next, the authors focused on identifying circuit composition, so specifically looking at which neurons are actually important in this overall network. The first way this was done was by looking at the computational model, right, and focusing on those sugar gustatory receptor neurons. And by activating these neurons at different firing rates, they were able to observe what downstream neurons were essentially activated and what firing rates they had. And this chart here, specifically part D, shows how the top 200 neurons were activated at various firing rates for these sugar gustatory receptor neurons. And then this basically is allowed to see which neurons are important in this type of network and which neurons are downstream and which ones are being activated by the sugar neurons specifically, as opposed to water or any other sort of gustatory sense. Then the authors used these types of neurons to predict which ones are actually activating motor neuron 9. And by focusing on that, we were able to see that there's a smaller section of these neurons that lead to overall motor movement with the proboscis towards the sugar. And then the study essentially took out all of these 200 neurons and one by one eliminated them to see which ones were necessary or sufficient for overall activation of motor neuron 9 and whether or not it would actually predict that properly. So we see that this allowed for, to see which neurons are actually critical in this type of network, and completely in a computational manner. And because it's done on such a large scale, it's more efficient than optogenetics, in which case you might have to go neuron by neuron or something like that. Overall, this allows you to see that there's a set of neurons that actually activate motor neuron 9, and those are a total of 47 neurons. But specifically, as we saw in part F of that previous graph, this one right here on the right, that certain neurons, when you eliminate them, actually not maybe silence, but reduce overall motor neuron 9 activity by 20% or more. And so those ones were described as inhibitory neurons by the computational model. So there were 14 of those out of 47. Now, something important to mention is that there are a particular series of neurons here that may not entirely be accurate by the computational model. The first one is phantom, because phantom was predicted to be inhibitory, the strongest predicted inhibitor, but is intended to be inhibitory, but we were not able to see the actual inhibitory action of phantom, likely due to the fact that the downstream neurons it was going to, such as scapula and roundup, those ones are, the basal firing rate of these neurons is 0 in the computational model, which makes it difficult for actual inhibitory dynamics to be propagated through the network, which is a bit of a limitation of this particular model, but it's something that can be addressed later on by just adding a basal firing rate. The next point of note was the usnea neuron. So usnea was also important. It's been difficult to assess its overall impact on the network, but it's been postulated that it's inhibitory. And is it
all right if I ask you a question?
Yes, please.
I guess I'm just, I had quickly reread the paper yesterday and I might have misunderstood this, but there's something about the terminology of inhibitory and about neurons that decrease firing when silenced that is confusing me, because you would think that neurons that normally are excitatory, so that are causing firing, if they're silenced, that indicates that they had an influence on firing, so they're in a sense excitatory. Why are these being called inhibitory then? I'm just trying to understand that terminology.
Oh, sorry, I may have mixed up my phrasing. Yes, these neurons are excitatory. That's my bad. I'm sorry. I was referring to phantom and saying that those ones were inhibitory. But yes, these neurons. Yeah. Sorry about that. But yes, so phantom was described as inhibitory because it's, even though you would see it being excited, the connectome would predict that it's an excitatory neuron because it is excited and causes overall impact in the network. We've empirically seen that scapula and roundup are actually inhibitory. Scapula is excitatory and inhibiting. Roundup is inhibiting, but we don't see the inhibitory action of roundup in the actual network because that basal firing rate is 0, and that leads to a lack of actual inhibition in the neuron. So that's the first one, and then the other limitation is usnea, specifically because usnea has been postulated to have more specific impacts in terms of being neuropeptidergic, and the authors described this by actually doing a knockout study. They took out the Amontillado protein, and that led to an increase in firing rate. So there was a clear difference in this type of prediction as well, which was a limitation of the model because it can't predict, peptidergic or essentially anything that's not directly related to the firing of neurons throughout the network. The authors then verified all of these predictions from the computational model by actually doing an optogenetic study. And overall, this model did quite well. We saw an accuracy of about 71%, in which case the predicted rostrum ascension, the actual rostrum ascension, 71% of actual extensions were predicted by the model. And then there's like a 99% case in which predicted that it won't, and it doesn't actually extend. So overall, it's reasonably accurate in terms of prediction of these types of outputs. And this is a good case of this model because it's specifically focused on something that's entirely within the brain, limited complexity. So it's a good use case for this model. The next thing that the authors decided to do was to predict input circuit effects. And they focused on this one other gustatory receptor neuron called IR94E. IR94E is usually focused on low salt concentrations. And I think it's also been shown for some sort of mating purpose and for, I think, male exposure. But the actual impact of this neuron activation related to sugar gustatory receptor neurons was not known previously. And although it had been hypothesized that this was an excitatory they created another heat map by doing different activations of these receptor neurons inside the computational model. It was found that this is actually inhibitory, and it's not strong inhibitory in the sense that it's not completely eliminating the action of these sugar neurons. It's not completely causing that inhibitory reaction, but at slightly different concentrations of sucrose, we can see that it's pretty similar to what you would expect with a bitter neuron. And for context, when you have a bitter neuron that's activated at the same time as a sugar neuron, those effects do tend to cancel, and we don't have proboscis movement toward the sugar source because the bitter neurons are also activated. And we see that this is the case at both concentrations, at 50 millimolar as well as 1 molar of sucrose. But there was a slight difference between that and IR94 at 50 millimolar, the smaller concentration of sugar. And because of that, the authors decided to extend the study to 1 molar to see a higher concentration of sugar, to see if IR94E is really as strong of an inhibitor as the bitter neurons tend to be. And it was found that they aren't as strong. They do have some sort of inhibitory effect, but it's not a complete inhibitory effect. It's just partial. So we then see the authors describe this type of overall Venn diagram, showing that there are many neurons in this network, and they all tend to be compartmentalized, doing their own thing. There's some overlap between these neurons, and the biggest overlap we see is with sugar responsive and water responsive neurons. But other than that, we do see that there's a lot of separation in terms of bitter responsive, IR94E, and sugar responsive. So the authors decided to kind of extend this to focus on water responsive neurons to see what happens with the type of circuit as well. And so they essentially performed the same type of methodology as they did with sugar neurons by taking the computational model, increasing the firing rate of particular neurons, those receptor neurons for water, and then determining what sorts of neurons are firing after the water receptor neurons have been activated. And so the 200 most common firing neurons were shown. There were about 30 that were particularly strong. And then those neurons were taken to predict motor neuron 9 activation in terms of proboscis movement to the actual water. So we see that those a particular set of neurons are also activated. And then again, the study is done when you take out some of those 200 neurons, and one by one, you just eliminate those neurons, see what happens to the firing rate of MN9 by removing those neurons, whether or not it's activated at all. And then another heat map is generated in which you predict with sugar and water neurons to see whether motor neuron movement is happening or not. So overall, or I'll kind of discuss the summary of what the water neurons actually showed here, that there is overlap. There's significant overlap between the sugar neuron circuit and the water neuron circuit. And overall, those water neurons that are activated here, of these neurons, a large percent of them, I think about 30 of them, were actually used for the sugar neuron circuit as well, which indicates some overlap between that circuit, something that was shown by the computational model. I wanted to mention osmia again one more time, because it was kind of shown that in the sugar circuit, the osmia neuron was supposed to be inhibitory, but in this case, it might have some sort of excitatory or synergic effect. And this was kind of shown by taking the computational prediction and combining it with optogenetics one more time. So we have a series of flies, and you take those flies, you have that water neuron activation with optogenetics, and you kind of compare it with the prediction between that and the computational model. And we see that Bosnia actually does have kind of a synergic effect, that even though you think it's not required for water proboscis extension, they're still firing of that specific neuron. And that kind of just goes to show, again, that there is slightly more complexity to this network than predicted just by the computational model, because the computational model doesn't say that this is required, right? But we still see its effect in this type of system. So again, that's kind of a sign of how peptidergic effects, along with basal firing rate for the inhibitory neurons that I discussed previously, along with a host of other factors that I'll also discuss, may be used to add in some complexity to this type of network and allow for more insights on future study. And the final component of the study was to assess the Drosophila grooming circuit. So we've seen sensorimotor impacts with a smaller scale circuit in the Drosophila brain. But now we actually want to see how this impacts another one, which is grooming. So this kind of focuses on Johnston's organ, which is in the brain of the Drosophila. And these are Johnston's organ neurons related to the antenna. Or what happens is these sensory neurons can either communicate to interneurons, which are ABN1 and ABN2. And they also communicate to descending neurons. But we're not exactly sure which descending neurons the Johnston organ neurons communicate to. So it could be ADN1, it could be ADN2. We're just not entirely sure at the moment. And so this computational model was kind of used to assess what sorts of connectivity we have present in the circuit based on that. And downstream, this leads to antennal grooming in the Drosophila. So again, this type of study was done where you take all your Johnston organ neurons and you activate them, and you predict differing firing rates what sorts of neurons are activated. And you take the top 300 in this case of those neurons to assess what is going on. And based on those 300 neurons, when they were activated, we assessed what would happen to ADN1, which is that downstream neuron, and we would assess the firing rate of that neuron. So overall, we see that there's only a couple of neurons that are actually active that lead to this type of ADN1 activity. Those are ABN1, ADN2, and ADN1, which we also showed previously in this slide. So ABN1, ADN2, and so forth. And then again, that knockout was done or not really a knockout, but you just inactivate one neuron computationally and see what effect that has on ADN1 downstream. And so we see different firing rates for those as well. But really the most important ones with the solid lines almost that you can see are ABN1, ADN2, that interneuron and the other downstream neurons specifically. One other thing that was focused on here was that there's a large number of Johnston organ neurons, and there are different types of them. There's one coded CE and one coded F. And what the study did was they computationally analyzed what these neurons' activations would impact on ABN1 specifically, which is that interneuron. And we saw a population preference that when the CE neurons were active, they lead to a much higher firing rate for ABN1 as opposed to the F neurons being active. And this was also verified by actually analyzing calcium imaging and seeing that these neurons for CE activation leads to much greater activation of ABN1 as opposed to the F neurons. So we again see a match between computational study and calcium imaging, just some sort of empirical lab work that can kind of verify that these computational predictions are accurate. Overall, what the study did was it had stepwise model validation, right? We identified macro characteristics of the circuit, identified the key components of the circuit, like focusing on specific neurons, whether it be motor neuron 9 or gustatory receptor neuron for sugar, assessing the specificity of the model, seeing exactly what neurons are necessary or sufficient for network functionality, predicting the effects or non-effects of certain components, identifying circuit interactions. Then you go overall to unknown circuit outputs, such as the ones that we did with the IFR neuron, and then focusing on unknown circuit characteristics, larger unknown circuits, and then completely unrelated networks. So essentially steps 7 to 9 are kind of future steps that this study and other studies are actively working towards identifying, right? And something that I think is a goal of the Carboncopies Foundation as well, in terms of creating larger scale emulations that could be used with some of the results that we've learned in a study like this. Now, the authors made some notes about this model that could be improved. So as mentioned, they're exclusively excitatory or inhibitory neuron dynamics, specifically in terms of, that it can either cause something to fire more or fire less. The absolute firing rates are approximate, and they're not exactly determined empirically. And morphology, gap junctions, exact synapses, or neuromodulation were not accounted for in this type of model because the leaky integrate and fire model is quite simple and doesn't really have that level of complexity to show all of these factors. And excluding the optogenetic split GAL4 experiments, which focused on the sugar validation using optogenetics, there's 84% accuracy overall of this model, which is reasonably good in terms of assessing the efficiency of a computational model, especially one of an entire Drosophila brain, because this is essentially unprecedented. We haven't seen anything like this before. One other thing important to note is that these types of dynamics have also been seen with prior brain emulations such as, or not brain emulations, but connectomes like C. elegans. But it's been shown through calcium imaging that these types of interneuron electrical circuits are not 100% predictors of exactly what goes on in a C. elegans brain. And there's a particular study I found that I can also share the link of later that kind of shows how there's more complexity to this than simply identifying a connectome, even though connectome does reveal a lot of information about what's going on. So future steps may be to have more sophisticated neurotransmitter or receptor information, having a non-zero basal firing rate to better capture inhibitory dynamics, which is something we specifically saw with the phantom neuron earlier, combining it with other computational models for extrasynaptic signaling, which is something that's been seen with C. elegans before as well, specifically like what data cannot be represented in a connectome, and identifying that and trying to add that onto a model like this. And then focusing on sensorimotor dynamics involving descending neurons, which are not complete in Flywire, especially the ones related to spinal tract and so forth. Maybe not in Drosophila, but in future animals. So conclusions of the study were that a simple leaky integrated fire model, when constrained by connectivity and neurotransmitter information from the connectome, can accurately predict circuit mechanisms underlying sensorimotor transformation. And computational predictions were verified by optogenetic experiments, as well as that one calcium imaging study. So IR94E neurons, which were previously thought to be attractive, were shown to be aversive and inhibit proboscis extension, which is a unique feature of this computational model in what it showed, because this was not previously, shown by lab studies. Then the sugar and water taste processing pathways were shown to share a common set of neurons, which forms that consumption circuit, that gustatory circuit. The model can be applied to different sensorimotor circuits, such as the antennal grooming one, which shows the versatility of this model. And further refinements with slightly more detail, as mentioned earlier, will likely improve model accuracy, which can add significant value to studies like this and to neuroscience overall. So that was overall the Journal Club, and I'd love to open it up for discussion.
Thank you very much, Archit. And I don't necessarily want to be the first person to ask questions because, that's a tendency of what tends to happen. But I'm going to make a tiny, tiny exception because there's something specific I wanted to do. And this is because, of course, when we're thinking about functionalization and brain emulation, all of that, our main concern is about how you actually carry out the parameterization of the model. And so there are some really simple things that I just wanted to check with you first. So throughout the presentation, when you're talking about cells, are you talking about individual cells or cell type?
So specifically in this case, I believe it's the specific cells themselves, because we're focusing on the actual circuits. For example, with motor neuron 9, that's because of the lateralization. You see like a motor neuron 9 in the left hemisphere and the right hemisphere. Both of those are analyzed. So those are specific neurons. Even though they're both type, I guess their type is motor neuron 9. They're both specifically analyzed, one on each side. So. Okay.
I think in some cases that might not be the case, like with the I94E, those are probably cell type. Those are ones that get labeled that way. Okay. Yes. Then another thing I wanted to check is, so when you looked at the setup of the model, the methods, the whole connectome there was available, but of course, the question is how much of that was actually parameterized and included in the model? What do you know about that? Was it just a small portion here, or?
From what I've gathered, they attempted, so they had the entire connectome that was actually, I guess, used for simulations, but I don't recall significant. From what I recall, there was a portion of the paper that discussed how the circuits that were being analyzed had more detail than the rest of the model. But I don't exactly recall what detail that was. I do know that, like, obviously when we had certain neurons, like the sugar neurons that were fired or stimulated in the model, we analyzed all of the downstream neurons, right? Mm-hmm. But I don't think that all of these neurons had the exact amount of detail in terms of some specific parameters. I don't recall exactly, and I can definitely look into this. And
yeah, I'll mention
Yeah, go
if I
ahead.
may, that we basically treat all neurons identically. Uh, um, and like the only free parameter basically is the weight parameter, which is based on the connectome. We could have in principle like gotten more parameters from the literature, but we're not convinced necessarily that we did have the ability to determine if that actually improved the model accuracy, if that makes sense, because we don't have good um, electrophysiology.
Thanks, Phil. And that free parameter, the weight, that was obtained through synapse counting?
Yes,
My knowledge,
exactly.
Yeah. Okay. And so one of the things I'm super curious about in this model is, well, actually, there are two things I was wondering. One, I'll ask a simple question first. In what we might call the knockout study, where you're taking out one neuron at a time, I was wondering exactly how that was done and why knocking out one neuron at a time, or instead of, say, for example, trying combinatorial possibilities of multiple neurons being knocked out as, I don't know, pairs, groups, whatever.
yeah. So based on what I read, I believe the purpose of this was to identify necessary versus sufficient for certain types of downstream firing. So they just wanted to go one by one to identify which neurons are actually critical to firing and how they are actually affecting that firing. But I know that based on network dynamics, there are obviously combinations of these neurons that could have further elucidation on that front.
If you're just knocking out individual neurons, that almost assumes that they must have kind of an overpowering influence, right? It's sort of that they're sort of absolutely central. But maybe that was the reasoning.
Yeah, I guess sort of the reason was that it was something that we could verify empirically. And we have done definitely we tried triple or quadruple knockout silencing computational silencing experiments. Like we did it singly because we could compare it against our single not silent optogenetic silencing experiments.
Awesome. Okay, so here's my other, I don't know if it's really a bigger question, but the question that's more about why I was asking about the whole model and say what was actually parameterized into what detail. And it's because of the question of the influence that the rest of that fruit fly brain has on this circuit, or can have, if those parts are also in some way active. For example, if you have spontaneous activity or random activations in there, which also might then offer an opportunity for not having a baseline 0 hertz in a sense. So I'm wondering what thoughts anyone might have about that, about what happens if you have not just the circuit in isolation being activated, but if you have the rest of the model around it, say the rest of the brain, and if you have, for example, some kind of randomized firing going on. Or maybe the question is too complicated or not clear.
I know that, I guess in this model, this wasn't shown, but I know it's been a pretty big point of focus for later studies as well, just adding in that randomization. I know computationally, this may be a little bit more expensive, and I'm not sure if that's a factor at all, but I suppose Dr. Shiu will know if that's a case of simplicity or just because it's not as necessary.
Yeah, it was computationally more efficient and but one thing we think we could do with this is that with something like if you think about Maslow's hierarchy of needs, given the fly being in a particular internal state such as hungry or fearful or something, we expect that it would change, say, the fly's willingness to eat or something. And in some cases, we have seen that and wasn't in the paper, but I think that's exactly right. That there are a variety of internal states and other computations in the brain that would definitely influence the circuit.
Cool. Yeah, because that was something that I was also wondering about with some proposals for experiments in mice, where you could say observe a small patch of the cortex and and use those activity recordings for information about reconstructing connectome, but then you have the entire brain of the mouse underneath that influencing what's going on in that patch and which things are actually being expressed at that moment, which activity is being expressed in this little patch you're watching. So I guess that's one of the things I was just wondering about in terms of methods and approaches. Before I ask any more questions, because of course I have way too many, I'd love to give other people a chance. And feel free to just speak up without raising your hands.
I have a really basic question, which is, is it true that we have now gone farther in brain emulation of flies than we could in C. elegans? And if that's true, why is that?
Yes. So the C. elegans connectome was, at least based on what I've explored of it, it was very primitive in that you don't really see a lot of output. And essentially the interface that I had was that it was just sort of a simulation of this worm just moving around, and it wasn't actually accurate related to the dynamics going on inside the neuron. It only has 302 neurons, so it's not as complicated. Based on this, we're able to actually see the downstream effects of more complex stuff like sensory dynamics or antennal movement like we see in the fly. So it allows for a lot more understanding of what's actually going on in terms of complexity with something like this.
And the second part was, why have we gotten so much further in flies than worms?
I can try to address this. This is something that's puzzled us as well. I would say one of the main things is that in C. elegans, our guess right now is that the fact that there's only 302 neurons means that each neuron needs to be doing a bunch of different things. And so a lot of their circuits work in context-dependent manners. And it means, and there's also all this like extrasynaptic communication. And they're not really like, they don't fire action potentials per se. So at least almost all of them don't fire action potentials. And so they're a little bit weird. And so I have a bunch of opinions on that, but those are my general thoughts. But I'm not fully a worm neuroscientist, so I don't fully actually have a great answer.
Yeah, if I can jump in with a controversial opinion, I would say that this, there have been some papers recently that basically say you have to describe a neuron as an entire neural network itself, because there are so many details that you have to take into account. Then my controversial opinion would be, yes, this is more true for C. elegans than for something like Drosophila or mammal, where the population matters more than the individual neuron most of the time, whereas in a C. elegans, the individual neuron matters completely. Yeah.
Can I ask a follow-up question to this? I'm actually very curious. Do you know if there's been any work on evolutionary changes, I guess, that certain neurons become less multi-purpose, or how that changed over time? I guess I'm not sure if my point's coming across, but it's very different between.
I know what you're asking, but I am unfortunately the wrong person to ask. So maybe someone else here knows this. I'm not really well versed in the evolutionary track. Sounds like perhaps no one here knows that, but it's interesting. Yeah. But then, of course, you'd expect that what you'll find is that perhaps the older cells in the mammalian brain would exhibit more of what you'd find in more primitive animals, perhaps going all the way down to C. elegans. I don't know if we can trace it all the way that way, but it's an interesting question. Yeah. Are there more questions before I start stampeding again with other questions?
Hey, Archit, I have a question. I think it connects to what was just been discussed. You mentioned that you've looked into some literature on what parameters cannot be estimated from the electron microscopy connectome on C. elegans particularly. So maybe you can tell us a bit more about those and I will then have some follow-up questions probably.
Yeah, sure. I'm not an expert in C. elegans specifically, but based on what I've at least read, and I know this was actually in the partially mentioned in the paper as well that I described, that there's some specific activity propagation that's been studied with C. elegans, and it's been shown in calcium imaging and other studies that isn't directly predictable from the C. elegans connectome or other sorts of dynamics in that sense. And I know I briefly saw one paper related to this that described that overall, but I'm not entirely sure what that means and why certain extracellular signalings are more significant as opposed to others. I know it's a topic of ongoing research as well, but I'm not, I confess I'm not perfectly up to date on exactly what's going on with that.
So maybe I can then open up the question slightly and ask whether, if there's anybody who knows the most specifics, do they also believe that the fly connectome would not be susceptible to that lack of information?
I certainly can't answer that with any kind of certainty, but I think it would be surprising if you didn't find that there were at least some places in, say, a Drosophila brain where these bits of missing information would matter. That's just my guess, I guess. Yeah.
I know there have been some studies related to fields of how certain electrical fields can impact signaling, but I can't point to a specific study that would be relevant to this discussion, but I can definitely look into this as well.
I would like to take a moment to read out some questions that Alex McLean put in the chat. So his first question, well, he said that I partly answered it, but he was wondering regarding measuring neuron weights by counting synapses. I was wondering if their postsynaptic density was also accounted for, or if it's just counting number of synapses present at an individual connection. That's his first question.
It was purely number. The numbers are pretty high, and so I think it may that's something that could be done, like in terms of maybe larger synapses or whatever should be weighted higher. And it's something we considered but never did. Certainly it could be possible, but it didn't seem to be necessary.
Thanks. His second question is, were the biological parameters mapped to the leaky and integrate model by hand, or was it done automatically?
So the biological parameters were identical for all neurons, and so we just selected them from the literature. So the biological parameters might be the resistance or capacitance of each of every neuron in this case, but it's just
right.
a single parameter that we chose that has been measured electrophysiologically.
Yeah.
From
so then I guess because it's only one set, it's duplicated across all of them. Hence, in a sense, that's automatic. Yeah.
Yeah.
All right. And then his third question, final question is, will or have there been attempts to redo the existing work with other types of models, such as Itzkevich, and comparing with the leak and integrate to see how much they differ?
Yeah, that's a great question. We never did. So we did something kind of similar, which is it's modeling them as dynamic equations, just as ordinary differential equations, and our model performed a little bit better than that modeling, but not with the other types of neuron modeling. We weren't convinced we had enough resolution in terms of ideally what you would want is the firing rates of say MN9 given some particular input, but we just don't have good electrophysiology data in Drosophila to do these really high resolution experiments to compare individual models at a really high resolution.
Thanks, that's great. More questions?
Dr. Shiu, can I ask you a question? So I was looking through the code a little bit. I know you use the Brian 2 library for computing. Can I ask how computationally expensive this was, and what would it look like to scale it up, I guess, if you wanted to use do you need to use a different library? And if so, I guess what would it take to just change the type of neuron to something else?
Yeah, so I can run this on my laptop, and my intuition basically is that it's not especially computationally expensive. It's not, but it's also not computationally efficient either, because this currently runs on CPUs, and like if there's ways to get it to run on GPUs, but we didn't. For our purposes, it didn't work especially well. So like once we can get it onto GPUs, then it can be super efficient. But it was not especially fast per se.
Makes sense. Thank you. Also, this may be completely wrong, but do you think there's a way of taking that model, adding in the random like a higher firing rate essentially for all the neurons, and then just giving it a large amount of sensory stimulation and seeing where it goes? Just is there a way to just log that data and analyze if it's any way similar to biological brain of a Drosophila? Because I know nowadays we have ways of just feeding in world models using AI generated data. And I was just wondering if that's something that's been on the docket.
Yeah, we haven't done anything like that, but I think that would make a lot of sense to like start with some random basal firing rate and feed in information. I think that would make a lot of sense.
Thanks. Anyone else have a question before I go again? Sounds like no. Or maybe I saw Andy just move towards the screen. Is that a no? Okay. All right. Yeah, so I can't seem to drag myself away from worrying about or having questions about when you're trying to functionalize or parameterize, and then you want to see how your model works, how much the working model, if we go beyond maybe this small set of limited tests that were carried out for very specific purpose, for very specific feeding and grooming behavior, how much it depends on what is constraining what I would call the neural code. So the way that neurons in the networks are talking to each other. And for example, again, here, I'm not a Drosophila expert. In the mammalian brain, it's very typical that that the communication between regions or even within a region are all carried out in the presence of, say, rhythmic modulation of a population. And of course, there's a recurrent inhibition. Now, recurrent inhibition, if that's present, that would probably be in here as well, so that's fine. But these sorts of things, you know, these constraints, how much was there an attempt to bring in anything that we know about those kinds of constraints in the way that in Drosophila, the system talks to itself, to constrain what's going on, to help with robustness of the system? What potential risks might there be or issues to deal with?
Yeah, I think that's a great question. We basically did nothing to deal with that. I think there's some, yeah, certainly some concerns about that. But like to some extent, it's a tool for like predicting neural activity given some input or some silencing. And in that regard, we weren't necessarily worried about that, but like certainly thinking about that would make the entire brain models more accurate.
Right. Yeah. I understand, of course, what the limitations were and also the aims of the study in that sense, but I guess I can't help wondering about this because, of course, we're thinking about steps towards what do you do to really emulate an entire circuit or system or brain. And so I wonder about the next steps and what we'll have to do and where things might break down. That's where those questions are coming from. And I had a recent conversation with Conrad Cording where he was adamant that we had to be extremely careful about the inverse problem in system identification. So this notion that no matter whether we're working in C elegance and we want to take a really high resolution approach where you put basically you're assuming scale separation at a level that is around molecular interactions, or we're looking at Drosophila or mammalian brains and we're happy with neurons communicating by spikes and basically looking at electrophysiology for the functionalization, that you have to be very careful about the fact that anytime you're putting a black box around something and saying this is the system we're identifying, so we're looking at input output there, that really what we're trying to do is because we're reverse engineering, is we're trying to do the inverse of that IO function. And that if you're doing an inverse, say you have your matrices that are explaining what the IO function is, you do an SVD, and then your eigenvalues, the ones that previously were the tiniest eigenvalues, the ones that are most subject to noise, variation, and so forth, if you're inverting them, they become the largest ones. And so you have to be much more careful about all the variations that can happen. Now, personally, I do think this is where constraints start to play a really major role. But I'm just wondering if anybody here has thoughts about that problem when trying to do this kind of reverse engineering. And yes, this is a bit out of left field.
I wonder, is there a way to have those parameters trained rather than having to do all of this mathematical wizardry, I guess, instead of doing that, just taking some sort of structure that we already have, like this sort of network, and just exposing it to a large amount of data and just training it and seeing where it lands.
So that relates back a little bit to, for example, the Lapalainen et al. Paper, the one about the connectome constrained networks, where they used the visual task to train the parameters that were remaining. Yeah, when you can do that, perhaps that could work. But it's really the amount of data you need to get it right that's the issue when you're doing this inverse problem, right? Because how much data do you need to use? How many possible input-output connections or input-output patterns do you have to test? And we're talking about spatio-temporal patterns and not just spatial patterns, because there's a little memory in the system as well. Anyway, now I'm wandering into the territory of things to worry about. Just wondering if anyone's thought about it.
So why are you worried? I'm not understanding why you're worried per se. It seems like
I am just worried because this is worried is maybe the wrong word. I'm thinking about it because this is one of the big arguments that Conrad is giving about going deeper and deeper, getting down to a much higher resolution when thinking about the problem. Because basically what he's saying is that at a very high resolution, when you're looking at just simple individual molecular interactions or something like that, that then the black box that you're trying to study is smaller. And so predicting everything it can do, in a sense, when you're trying to reverse engineer, isn't as full of uncertainty when you're doing this inverse problem. So it's kind of a question of how deep do you need to go? How big is your problem of data collection going to be? Because you're trying to prevent the problem of guessing or missing parts of your IO problem because you don't have enough data to match it. His comment is basically along the lines of eFIS is not enough.
Well, I guess it seems like it was enough in this study, though, right? Like it just was. You
For the
just do a
purpose
bunch
of
of
the
these
study.
experiments
Sure.
and see if it works for a given thing.
Yeah.
Yeah, I'm very skeptical of this idea that we just have to get all this complexity. It's like, you don't necessarily need to.
But also, isn't the purpose of an emulation to overfit your model to a human brain anyway? In which case, isn't it better to have as much data as possible? Because there isn't going to be as much variability between human to human on exact molecules within a particular neuron or synapse, right? But if we wanted to just have something that functions like a brain, then you could just train, I don't know, a transformer or some sort of other machine learning model, I guess. I mean, obviously there's a line between how much complexity you want, but I guess it makes sense that you would want as much detail as possible on a molecular level, right? If you're trying to emulate exactly what one human's brain is. I don't know if that's right, but intuition-wise.
I think you're asking the right question, and I suspect that there is a middle ground there somewhere. I think that this is, again, where I keep coming back to this notion of constraints. Like if you're really, if we're dealing in some parts of the brain where you're dealing with population activity as the main thrust of what's going on, and you have constraints on those so that you don't have as much variability as would naturally be present when you're looking at just a single neuron operating by itself, that this is already making the system more robust, and you're probably going to be able to avoid much of this problem that Conrad's talking about. And then there might be some cases where you're running into a situation where you do have to care because it's not part of the system that is so, population coding specialized. But again, this is still, for me, very open question. I'm just thinking ahead to the workshop that we're about to announce, where we're going to do, we're going to invite a bunch of people, including you, Phil, to talk about functionalization and ground truthing and whether or not there's a utility in using artificial data sets for that. And since Conrad is one of the people who's going to be presenting there, obviously this question is going to come up. So I know that we're at the top of the hour, which means that technically we're at the end, but I do want to make sure that anyone who still has burning questions can try to get them in. If there aren't, then I would like to thank both Phil for being here and being able to answer questions directly, and of course Archit for doing a great job presenting the paper, its setup, and the scientific results that came out of it. It's a fantastic paper, and thanks all for being here. Bye, everyone.
Thank you so much.
Bye, everyone.
Thank you for presenting this. Great.
Bye-bye.